Now look at this.
What's up, everybody? Today we do 5.3 on Mendelian genetics. Before we get into genetics, let's do a quick detour into evolution. There's a couple things that are showing common ancestry between all organisms. The first thing is that we all use DNA and RNA. So that's one thing that shows common ancestry. Another thing that shows common ancestry is that we all have ribosomes. And so you can use that genetic information to code for the exact same polypeptides. So you can take a human insulin gene, put it in a prokaryote, and it can now synthesize human insulin for us. And the last piece of common ancestry we see is that we all go through glycolysis. So we all have cytoplasm and we're all able to generate ATP using glycolysis. So when we look at genetics, we kind of need to go back into meiosis and think about our gamete formation. So there's two main laws we look at here. The law of independent assortment. As we said before, during metaphase, the homologous chromosomes are able to independently align on the metaphase plate. And law of segregation. Law of segregation says that our homologous chromosomes are going to separate from one another, which takes place during anaphase 1. So based on Mendel's laws, what would you expect from a monohybrid cross? Here we made a monohybrid cross. You can see that both parents are heterozygous for one trait. So we're going to pull down the allele from the male, the sperm, and pull across the genes that we find in the egg. So you can see that from a monohybrid cross is a 3-to-1 ratio of the dominant trait to the recessive trait. So every teacher is a little different. And so I believe in using the rules of probability and doing multiplication when we do some of our bigger Punnett squares. So what would we expect from a dihybrid cross? Here you have two individuals and they're heterozygous for two different alleles. So as you see on the top, we've got a capital R and a lowercase r and a capital S and a lowercase s, and same thing on the side of our Punnett squares. You'll fill the Punnett square in the same way we did the last one. And you notice that we have a 3/4 chance of the dominant phenotype and a 1/4 chance of each of the recessive phenotypes. But we're not done with our math yet. So we're going to multiply. If you have a 3/4 chance dominant R and a 3/4 chance dominant S, you'll multiply 3/4 times 3/4, getting 9/16. Same thing here with 3/4 times 1/4, giving 3/16. 1/4 times 3/4, giving you 3/16. And 1/4 times 1/4, giving you 1/16. This gives a 9-to-3 to 3-to-1 ratio with a dihybrid cross. So now let's make it harder. What would be the probability of getting this genotype in a tetrahybrid cross? Well, we know there's a 1/2 chance of getting heterozygous, another 1/2 chance of getting the other heterozygous, a 1/4 chance of getting homozygous dominant, and a 1/2 chance of getting heterozygous. So what's 1/2 times 1/2 times 1/4 times 1/2? 132. Stay tuned for part 2. If you have any interest, bye y'all.